This program is to accompany Chapter 16 in the Biology 2E textbook, which talks about gene expression. And so I'm going to mostly talk about gene expression in prokaryotes, but I'll also make reference to eukaryotes and some additional levels of control. And so we're going to talk about how proteins can turn genes on and off. And so we'll talk about DNA binding proteins that work in the major groove of DNA. In particular, we'll talk about one example of a set of genes that are controlled in prokaryotes, the lac operon. And in particular, proteins that are repressors and inducers work on this lac operon and to turn genes on and off. And this was the first kind of system where we actually got to be able to control genes, understood it. And so it was through the control of transcription. And this has had big implications for how eukaryotes do things. And that's a picture that right there you see is of a eukaryotic embryo of a complex multicellular organism. So just to remind you that genes are made into proteins. The genes are the stored genetic information in an organism, and they go through a two-step process, transcription and translation, and that makes them into a protein. And in the steps of transcription, the enzyme is RNA polymerase that is responsible for starting this process and making the messenger RNA, which then goes into translation. And there the ribosomes and transfer RNAs bring the correct amino acids to the so that they can be assembled into proteins, and those are the traits. And each of these things has an initiation, elongation, and termination step. We've talked about some differences in eukaryotes between how they make a protein. Prokaryotes, they group related genes into structures called operons, where if you have several genes needed to run a metabolic pathway, those genes will all be regulated together. And the transcription and translation will be linked, so they're going to be kind of regulated at the transcriptional level. In eukaryotes, it's a little more complex. That they almost never group related genes together, that a gene is by itself made into messenger RNA, and it's more complex. They have three different RNA polymerases that are used for different purposes. Genes have regions on them that are never made into proteins, that are cut out during this splicing of introns and exons. And then the messenger RNAs are modified so that to add information to them and target them and determine how long they're going to be active. So we're going to talk about both prokaryotes and eukaryotes here, but I'll do more eukaryotes later. Now we're mostly interested in gene expression in prokaryotes. And in here, almost all genes are controlled at the level of transcription. That's where gene expression happens. And there's a whole set of proteins that are regulatory that determine when a gene is going to be on and off. And these all bind to the DNA, and they bind at specific sequences that they access through the major grooves. And these proteins can be recognized by having particular structures that tend to bind DNA, and depending on their what the regions they recognize, they may block transcription or they may enhance it, stimulate it. This turns out to be the general model for all cells, and so it's worth talking about it in the simplest case, which is prokaryotes. And so this was all work done first in E. coli, and it's was done in the 60s and 70s when they were first identifying how things happened. So it turns out these proteins, these regulatory proteins, they all have regions that you can identify that they help them bind to DNA. These regions typically are going to uh, read specific sequences, and they'll get to them by looking at the major groove. And so the two major, well, there's two major types, but there's others as well. The two major types are the helix turn helix motif, where you have these alpha helixes arranged at a particular angle. And so those are shown in the upper picture. You also have a zinc finger motif that is going to work by having zinc containing proteins that attack. And then you have these leucine zipper ones, where again you have a helical region that picks up specific sequences by having this region here. And so these can read specific regions of a DNA molecule, specific sequences, and so they can identify out of the whole DNA molecule certain regions that they have to work on and bind to. These were first identified with the lac operon, which is a group of related genes that are all needed to use lactose as a source of energy in E. coli. And so lactose is an unusual sugar. You find it in the milk of mammals, and that's about it. It's a disaccharide that needs to be first split into two monosaccharide units, and then worked on a specific way so that it can be used as a source of energy. Most often, cells only use lactose as a source of energy if they live in a mammal and glucose isn't available. And so here there are three genes that are needed to use this as a source of energy: beta-galactosidase, permease, and transacetylase. They are all regulated together. And so this picture on the bottom shows the structural gene or the coding regions for them. And then you have the actual DNA sequences that are going to be turned into a protein. These are Z, Y, and A here. And then you have anterior to this, kind of upstream, you have regulatory regions. These regulatory regions include the promoter, which we've talked about before, but it also includes a structural gene for a repressor protein, a binding site for another protein called the cap binding site, and then a gene called the operon, as well as the promoter gene, or the operator. Okay, and so the operator and cap binding sites are recognized by DNA binding proteins, and there are two promoters here. One promoter is essentially always turned on. That's the promoter for the repressor protein. And so the cell is always making the repressor. Okay, and this operon or group of genes is normally negatively regulated, where transcription is prevented by the repressor protein, which the cell always makes. And what the repressor does is it binds to the operator site, and by doing so, it blocks RNA polymerase from reaching the structural genes. And so it's unless the inducer is present, this gene is always turned off. Okay. There's also positive regulation, but it's the negative regulation that really matters. The positive regulator actually, what this does is helps RNA polymerase under certain conditions. So let's look at this. Okay, so here's the structural genes we want to make: Z, Y, and A. They're at the end. Okay, and normally what happens is RNA polymerase binds the promoter, but can't make that Z, Y, and A because the repressor is in the way. The repressor blocks RNA polymerase from moving downstream to copying these genes. Okay. And so this is how the gene is turned off. And this generally happens when glucose is present. Uh, there is no inducer, which I'll talk about in a minute, and the promoter is just not all that active. Okay, so this is the normal condition, but the lac operon can be turned on, and this happens when glucose is kind of absent, and so that means that some molecules that are present only when the cell is low on energy are present, um, and so that inducer is going to be present when glucose is absent, and that will allow uh, a positive regulator, which is the catabolic activator protein, or CAP, to bind with these low energy compounds, which are cyclic AMP, and that will help RNA polymerase bind. And then the other thing that has to happen is lactose has to be present. So let's look at a picture of this with the operon turned on. Okay, so here's the enhancer. Okay, that's the CAP protein that binds to our DNA. And when it does this if cyclic AMP is present. That's ATP has been degraded to cyclic AMP when there's no energy. So this causes this CAP protein to bind to the DNA. That helps RNA polymerase. Okay, and then the other really important thing has to happen is that's the inducer. Lactose has to be present, and lactose binds to the repressor, which causes it to release the operator. So RNA polymerase is bound to the promoter and helped by the CAP, and it's no longer blocked. And so the cell makes the RNA for these genes that are needed to digest lactose. Okay, and so lactose can be used as a source of energy when these are made into proteins. And that only happens when lactose is present and the cell is low on energy. So probably not much glucose is there. Okay, and so this is the typical thing that we see in prokaryotes, that repressors bind to an operator that can block transcription and negative regulation, and inducers can allow that repressor to release the operator so that RNA polymerase can proceed, and then enhancers help RNA polymerase find the gene, and they can also be enhanced by the right kind of conditions. Okay, so later we're going to talk about gene regulation in eukaryotes. And generally here, there's more transcription factors that are both positive and negative regulators. There's also RNA editing and the transport of RNAs through the nuclear membrane. Okay, these things can also be partially controlled by small RNAs that can enhance or retard those activities. And then these messenger RNAs can be degraded over time, as well as the proteins. And we're also going to talk about a more recent discovery whereby we can have epigenetic modifications that affect things. But the basic kind of regulation we see in eukaryotes is similar to what we see in prokaryotes. And a really good example of this are the Hox genes. These are the genes in animals that control the development of the multicellular animals. They tell embryonic cells what genes to turn on or turn off. And these Hox genes are activated by DNA binding proteins that serve as transcription factors. And one Hox gene will switch on other groups of genes, and they're involved in the development of regions like the head or limbs or organs. And so we can turn them on and have different structures develop into different things. And again, these genes share promoters. And so when they share promoters, they can all be turned on at the same time. So I have a nice little video of that you can watch. It's only a few minutes, but it talks about the discovery of the Hox genes and how they're active. And I'll go through more examples of how eukaryotes control gene regulation in the next lecture or two.